In this video, we want to continue our discussion on the regulation of glycolysis and gluconeogenesis. So if we recall, we have the two pathways, glycolysis, where we go from glucose to pyruvate, and gluconeogenesis, where we go from pyruvate and form glucose. And there are three major regulatory points for these two pathways. The first one occurs here, where glucose is being converted to glucose 6-phosphate and vice versa for gluconeogenesis, where fructose 6-phosphate is converted to fructose 1,6-bisphosphate in glycolysis and vice versa for gluconeogenesis, and finally where phosphoenol pyruvate is converted to pyruvate in glycolysis and pyruvate is converted to oxaloacetate and then to phosphoenol pyruvate in gluconeogenesis. So these are the three major regulatory points that control the reciprocal regulation. Remember, these two pathways, we want them to be reciprocally regulated in order to prevent feudal cycles where we're producing a product and at the same time we're breaking that product right back down. So we need them to be reciprocally regulated. We've already talked about the major regulatory point, which was the phosphofructokinase 1 and fructose 1,6-bisphosphatase cycle or step, if you will. We want to continue and talk about the other two, and we want to start by talking about the regulation at this point here, where we have pyruvate kinase, and in glycolysis and in gluconeogenesis, we have the enzyme pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So let's look at the regulation of this key point in our two pathways. And we want to start by talking about the enzyme pyruvate kinase, which of course is in glycolysis, and it converts phosphoenol pyruvate into pyruvate. And therefore, since it's the very last step in glycolysis, we say it controls the outflow from glycolysis. Now, this enzyme is allosterically inhibited by ATP. It is allosterically inhibited by alanine, the amino acid alanine, and by acetyl-CoA. It's inhibited. Now, we, of course, know what ATP and alanine are. We'll talk about acetyl-CoA more when we talk about the citric acid cycle. But just suffice to say, when all of these molecules are at high concentrations, that tends to tell the cell that we have a lot of energy. Of course, ATP, we've got a lot of amino acids, so we don't need to make any more. And acetyl-CoA is also a high energy compound that tells the cell we've got plenty of flux through the citric acid cycle. So when these compounds are very high, it tells the cell we have a high energy charge and we want to shut down glycolysis. We don't need to keep producing more energy. And of course, glycolysis is an energy producing pathway. Alanine is not only amino acid, but it's produced from pyruvate in a different pathway. So pyruvate can be, through a different pathway, can be used as a precursor for alanine. And as I mentioned, acetyl-CoA, we'll look at that in more detail, but it basically is a molecule that enters into the citric acid cycle. Another thing about pyruvate kinase is that it is activated by fructose 1,6-bisphosphate. If we recall, fructose 1,6-bisphosphate is produced in the third step of glycolysis, and so this represents what is called a feed-forward activation. So here you can kind of see a diagram of glycolysis, and typically when glycolysis is turned off or inhibited, we start to build up fructose 6-phosphate. When fructose 6-phosphate levels get high enough, they feed forward and activate allosterically pyruvate kinase. Therefore, they help increase our flux through glycolysis. Here you can see an activity, pyruvate kinase activity versus its substrate, phosphoenol pyruvate, in the presence and absence of fructose 1,6-bisphosphate. So here you can see the curve to the right, the sigmoidal curve, is in the absence of fructose 1,6-bisphosphate. And we have less activity. We have a higher Km apparent. However, when fructose 1,6-bisphosphate is present, it allosterically binds to pyruvate kinase and our curve shifts to the left. Therefore, our Km is going to be smaller, giving us a higher affinity for that substrate. A little bit more about pyruvate kinase. Pyruvate kinase, kind of like phosphofructokinase 1, it needs to be its activity needs to be controlled not just by intracellular conditions, but also by what's going on outside of the cell. For example, blood glucose levels. So therefore, pyruvate kinase is inhibited by phosphorylation in the liver. So pyruvate kinase typically found in the liver is inhibited by phosphorylation. The kinase protein kinase A phosphorylates protein kinase, PKA phosphorylates pyruvate kinase and therefore inhibits it. It can also be dephosphorylated, so there's a phosphatase which removes that phosphate and makes it more active. Now remember, PKA activity is increased, so PKA is activated when blood glucose levels are low and we have an increase in glucagon. So glucagon binds to its receptor, if you recall. The receptor activates adenylate cyclase, which produces cyclic AMP, which activates PKA, and PKA therefore phosphorylates pyruvate kinase and inhibits it. So when blood glucose levels are low, what do we want? Do we want to keep using up glucose? No, we want to stop using glucose, stop glycolysis, and we want gluconeogenesis to occur. So when blood glucose levels are low, PKA gets activated via a pathway involving glucagon, and therefore glycolysis is inhibited at this step. And then, of course, as we mentioned, it can also be dephosphorylated, so it's not a permanent inactivation by a specific phosphatase. Here you can see a figure kind of illustrating that. We have phosphoenolpyruvate being converted into pyruvate by pyruvate kinase. So here's our enzyme pyruvate kinase. When blood glucose levels are low, then our enzyme is phosphorylated by PKA and we get the less active and pyruvate kinase, therefore lower glycolysis, less glycolysis. And then we have a phosphatase, which comes along when the conditions are right and dephosphorylates that, therefore increasing its activity and kind of turning glycolysis back on, at least at this regulatory point. Now the other enzymes in that pathway, the pyruvate carboxylase and phosphoenolpyruvate carboxykinase are not very highly regulated. They're more regulated at the transcriptional level and not so much allosterically. So that brings us to our last point of regulation, which is here where we have hexokinase and glucose 6-phosphatase. So let's look at the regulation of these. So if we look up here, here is glucose being converted to glucose 6-phosphate and vice versa in gluconeogenesis. And the regulation is pretty simple. Hexokinase is inhibited by the product. So as the product builds up, glucose 6-phosphate, it inhibits hexokinase. And as the product, or what would be the substrate for glucose 6-phosphatase builds up, it activates glucose 6-phosphatase. So that's really a very relatively simple regulation. It's basically controlled by the substrate or product concentration. So just a couple of more quick points about hexokinase. We've talked a little bit about this previously when we were discussing glycolysis. Hexokinase, its regulation is somewhat controlled by the expression of different isozymes, which of course are different forms of an enzyme. They have different amino acid sequences, but they control the same or catalyze the same reaction. These isozymes often have different KMs. An example of this would be the isozyme hexokinase 4, also called glucokinase, which is found in the liver. And of course, the liver, after a meal, after insulin binds to the liver cells, the liver can take up a lot of glucose all at once. Glucokinase has a very high Km. So when glucose levels are low, its activity is very low. It's not very active. But when glucose levels get high or they spike after a meal, glucokinase is able to bind the substrate and catalyze the reaction, converting it to glucose 6-phosphate. So it's active at very high or relatively high glucose levels. Hexokinase, as we mentioned before, is not allosterically controlled, and it is inhibited by the product glucose 6-phosphate. So that concludes our discussion of the major regulatory points of glycolysis and gluconeogenesis. So we certainly want to be familiar with how these important pathways are controlled.